Okay, so now we're going to move on to another group of biological molecules. We're going to look at lipids, and lipids are going to be really different than the other types of biological molecules, partly because they're called nonpolar, right? They are going to have a lot of carbons and hydrogens that share electrons equally. First, all the carbons share electrons equally among themselves. And then the hydrogens also share equally with carbon. So there are not going to be any charges or partial charges, or at least very few within these, which means they're not going to interact with water very well. One of the most common characteristics really of lipids in general is simply that they're nonpolar, or at least mostly nonpolar. Now, lipids are going to be different in another way than the other biological molecules in that there isn't a single type of small monomer unit that gets repeated into polymers. So in carbohydrates, you'll have simple sugars, and they'll form long chains of sugars. In proteins, you'll have amino acids, and then they'll form long chains into the complex polypeptides in proteins. DNA and RNA as well. They have a nucleotide that's a repeatable unit that then gets attached into long chains as well. But there's no such thing in lipids. The closest we have is something like this structure, which is already drawn up here because it would take a long time to put there. But we're going to break it down, look at the pieces of it and what's common that we do find in many and most lipids, and then how you would depict that in a drawing yourself and explain how the structure is put together. So with lipids, so now talk about them just where we see them. In the cell, lipids are going to be one of the main long-term energy storage molecules. In addition, they're going to be the main structure of the cell membrane. So we're going to be particularly focused on a group of lipids called the phospholipids, which we'll get into a little bit later. And they're going to follow the same basic structure here as this triglyceride that I have up here. But they're going to be structure all the outer cell membrane and the inner membranes of organelles in prokaryotic cells. They're not inner membranes, but the gram-negative cells have two phospholipid bilayers. So the phospholipids are going to become very important in cell structure, as well. So how do we break this down? What's the most simple unit? Well, one common unit we have for the lipids is a fatty acid. So a fatty acid, break down the term here. So the fatty part is really going to be a chain of carbons. So it's going to be carbon, 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 like this. Now these carbons, typically there's a range of numbers of carbons. So say 14 to 20 carbons we see in fatty acids. So you might see all different lengths of them. Typically not that many smaller than 14. We don't see that many bigger than 20. We can, but there's going to be these long chains of carbons. These are all non-polar covalent bonds between them. I'm not going to draw 20 because it just gets to be too many. The carbons are all going to have hydrogens attached to them. So for every carbon is going to have hydrogens attached to it. And typically the full number, and we're going to get into some variation within this, something called saturated and unsaturated. So that's the fatty part. They're going to be these long chains of carbons. The acid part. If you remember, what is an acid? Acidity is defined in a couple of ways, but in particular, we're talking about hydrogen ions when we talk about acidity and pH in general. And so acids were molecules that donate hydrogen ions into a solution. So it's something that's going to be giving up hydrogen ions. Well, one of the functional groups that you're going to need to be familiar with is called a carboxyl group. Right? And a carboxyl group can be written in a few ways. Sometimes we see it in type in a book, written like this, COOH, but that's not its actual structure. It's not how it's put together. You may also see it written as COO negative, like this, meaning that the hydrogen has been broken down and a proton or a hydrogen ion has gone out into solution and has created this other form. So we see it written in both of these ways. I'm going to draw it with the hydrogen here. So this is a carbon, and now these two O's, they're not actually connected in series. One of these is a double bond to an oxygen. The other is a single bond to an oxygen, like that. And then this oxygen is going to have a hydrogen attached to it, like that. So the carbon has its 4 bonds, 1, 2, 3, 4. The oxygen has 2. This oxygen has 2. The hydrogen has its 1 bond. That's the carboxyl group, and it behaves as a weak acid. Now, what does it mean, weak acid and strong acid? This gets back to another discussion of pH. Acids, which can give up, again, hydrogen ions to solution, can typically take them back up again, right? So a weak acid can bring back that hydrogen ion and give it to solution either way. A strong acid, when it donates the hydrogen ion, it doesn't take it back typically. It typically just gives it to the solution. So this is our carboxyl group, which acts as an acid. So we have the fatty part here and the acid part here. So this is where the name comes from. And that's our most simplistic unit of a lipid, is the fatty acid. But what we're going to find is that unlike the sugars, where we have a simple sugar attached to a sugar, fatty acids do not attach to other fatty acids. So you don't have fatty acid to fatty acid to fatty acid. They attach to some other molecule. Right? This molecule is called a glycerol. And we're going to see it and other molecules like it fairly often. Glycerol is a 3-carbon molecule. So you have the 1, 2, 3 carbons. Each carbon has an oxygen attached to it. This is sounding a little bit like a sugar, but you notice it's glycerol. All right, so that means it's an alcohol. It's lacking the carbon-oxygen double bond that we would find in a sugar. So it's not a sugar. It's technically an alcohol, all right, molecule, and it's what's going to be the joiner for the fatty acids. So they won't join to each other, but they'll join to the glycerol molecule. All right, and that's going to happen right here. So the glycerol molecule would normally, could draw it like this, have OH, OH, OH. Okay, so that would be a glycerol here with its OH hydroxyl groups. And then we would have our carboxyl group here. So this would actually have an OH, right? Like this. So triglyceride, 3 fatty acids. 3 fatty acids get joined together, but not to each other. They join to a glycerol, which has the 3 carbons and the 3 oxygens. The area where this is going to happen is right here. If you remember from when we looked at sugars and how sugars come together, we had something that I talked about called a condensation reaction. Condensation reaction. If you remember that word condensation, the thing that probably pops into your mind is water, right? So a condensation reaction is where a water molecule is formed and a new bond is formed. So to make water, we need 2 hydrogens. They come from here. 1 hydrogen, 2 hydrogens. And then we also need an oxygen. So we take away an oxygen as well, right? So we remove 2 hydrogens and an oxygen. And then during a condensation reaction, a new bond is formed. So here's the new bond. Okay, so that's the new bond that joins the fatty acid to the glycerol. And then this would repeat and repeat, and then we end up with the 3 of them together. And that's how we form a typical triglyceride. So the structure of a fatty acid is going to be a long chain of carbons all put together. Non-polar covalent bonds, equal sharing of electrons between the carbons. The hydrogens are going to bond to the carbons with non-polar covalent bonds. And so we have this very long non-polar section. So we got non-polar. Double it up here. Doesn't like water. Doesn't interact with water at all. And then we have the weak acidic carboxyl group. So these bonds here are going to be polar. That's polar, as well. And then it can tend to form a charge as well. So we have this little area here where the oxygens are, where there could be an interaction with water. But in general, doesn't like water, doesn't like water, doesn't like water for the most part. So this is when you take olive oil. Olive oil is a triglyceride. Mix olive oil with water, what happens? they don't mix. They don't mix together at all, because the water is the polar molecule with the partial charges, and the oil is non-polar and it doesn't have any charges, so it can't really interact with the water. Right. So that's the first part of this, is looking at the introduction to what is a triglyceride, what is a fatty acid. Second part is going to be to look at what we call saturated versus unsaturated fatty acids and how that affects the triglycerides. Then we'll move on to the phospholipids, which is just going to be a variation of this structure. And then finally sterols, which are a completely different class of lipid. So this is the introduction right now. Make sure from this what you know. You can identify a carboxyl group in type in writing and drawing. So be able to draw or sketch out a carboxyl group yourself. And then know and show where the fatty acid actually joins the glycerol. So make sure you know the glycerol and make sure you know how those things can come together. All right, so this is what you're going to need from this particular section of information.